Thanks to be here, and I'm really happy to present the work that we are doing on some of the stuff on phage and phage bacteria interactions. My lab particularly focuses on microbial interactions and understanding the mapping of these interactions so that we can start engineering some functional ecologies where these interactions matter in applications, whether it is soil or human health. Particularly, I'll be focusing here on the phage angle, as usual, right, in this conference. So this is one of the key knowledge gaps that we have in microbial ecology is what is a sequence to function relationship. So typical style is really isolate microbes from different ecosystems, sequence them, use computational models to predict what does the gene function structure look like, build some flux analysis models, and then we make a speculation of what must be happening in the nature. And one of the key gap here is the experimentally validated what is the function of these genes that these microbes encode. So for example, here, the blue is computationally predicted function, and the gray part is there is no comparison to any data that is out there. So you cannot predict any functions. Experimentally validated functions are in that green color pie. So this is from 1,000 bacterial strain published a decade ago. This has not improved any further. So in fact, this is severely compromised when you compare it to phages. This is the complexity we have. So phages, obviously, we know they are everywhere. So we have heard enough talks about phages present in every environment we are associated with. However, the studies that we have known so far are on model organisms and model bacteria-phage interactions, which is important, as we just heard from Kim's talk. Phage infects and bacteria respond. And this war has been going on for millions of years. So thankfully, untargeted metagenomics is giving a lot of diversity. What is the phage diversity looks like in the biosphere? This is the almost 5 years ago published paper from JGI showed what is the diversity looks like Earth virome, where they predicted phage-host interactions. But we know that this prediction of phage-host interactions is not really perfect, and we need a lot of studies on this. Obviously, the other knowledge gap in this is phage function, gene functions. So we don't have any systems understanding how do we associate the function to the genes. So obviously, it's the largest diversity of the functions, which we don't know more than if you isolate a phage in a lab, it's a high chance that 80 to 90% of the genes we do not make any, we don't have the functions annotated by using the tools we have. These unknown functions are challenging when you're trying to build ecosystem models, and beyond that, thinking of climate change and how phage is interacting soil health or human health and so on and so forth. Unless we understand those, at least to a level of sense, that it's going to be challenging. So really, it also limits how we use these phages for different applications. And that is one of the things that our lab works on. One example, concrete example, this is the Felix-01 phage, which actually in fact kills 98% of more than 98% of Salmonella species out there. And this is used for diagnostic purposes. But more than 60 to 70% of the genes of this phage, which is used in diagnostics, is we still do not understand. So there's a lot of studies we can do all lifelong. So what are the knowledge gaps that are driving our lab, particularly is understanding what are the different infection pathways in different model systems? Can we understand phage resistance mechanisms? If you want to understand it used for applications, we need to map that. Can we understand what is the phage host range looks like, or can we predict them? And also interesting is the cross resistance. How do I map these phages? When one phage infects, does the other phage is resistant to it? What are the trade-offs? What are the virulence factors, functional traits, or fitness, or a tolerance to a metal? It gives the sensitivity to phage? So all of these trade-offs are interesting to study. And then obviously, we need tools to engineer them so that we can study them, we can understand the functions of these phages, and then overall, how do they impact the microbiota? So my personal moonshot is, can I, given a bacterial genome sequence, can I predict a phage just based on the sequence? And that's a long shot. I don't think so we are anywhere near there, but it's something which I keep motivating myself towards. So today I'm just going to give you snippets of these three different things, focusing on phage-host interaction generation technologies that we have used so far, understanding the gene function, and then also understanding how do we rapidly start mapping gene essentiality in phages so that we can start understanding how do we make these engineering applications reality. So the first one, few years back now, we started developing these couple of technologies. I'm directly associated with a couple of them, but not all of them. So RB-TnSeq is a transposon mutagenesis, where insertion on a genome of a bacteria, you can have a barcode on it. I'll cover it in a minute. So essentially, once you map this barcode insertion on a genome, then you can use this for a variety of applications. A bunch of publications followed up on these technologies. CRISPR-I, I'm not going to spend much time on this. Essentially, inactive, catalytically inactive Cas systems can be used for gene knockdowns, and you can use that application for a variety of things, understanding the gene functions. And then the third thing is a DUP-seq, is essentially a overexpression library with the barcodes on them. So you take a bacterial genome, you chop it up, and you clone it in with the barcodes on them, and then you can use that as a pooled fashion. And this work is not my only lab. I collaborate with two labs here in Adam Arkin, Adam Doshvar. They are collaborating labs, and then a bunch of people listed here who have been really pushed the limits of these technologies. I'm going to touch upon a couple of these technologies in some detail, but one of the key things about these technologies is barcodes on them. So if you have a pooled library of barcodes, either it is a loss of function or gain of function, then you can expose them to different conditions, whether it is high metal, high salt, phage, antibiotics, you name it. You can take these 96 different conditions, and then you can essentially pool the survivors. If you add antibiotic, most of them will die, some of them will survive. Collect the survivors, collect the genetic material, and then you can do a simple barcode PCR, and then you collect that before and after, then quickly start understanding who survived and what do they encode in a function. And these are pretty economical. Each assay can cost you $10, and then you can really rapidly test this under different conditions. If you Google fitness browser, LBNL, this is the data that you will get. More than 70 to 80 different bacterial strains have been created a library. So you can take a library and then quickly put a phage of your choice and then identify the receptor that it binds to. So I'll touch upon some of the data here I'm showing, but it is a huge microbiology resource. If you're thinking of a milestone, this might be one of them in terms of microbiology. So the CRISPR-I, obviously, we can design CRISPR libraries. You can pull them on a chip. Twist can generate these libraries much cheaper now. You can put it in a strain of choice, and then you add a condition of interest, whether it is metal, phage, and then quickly see the survivors, and then start to addressing more screenings will give you more hypothesis-driven research later on. So the dual barcoded library or overexpression library starts with a simple, any DNA for that matter, whether it is a phage or a bacteria, you chop it into a small chunks, and then you clone it in a double barcoded library. These dual barcoded libraries here, you insert the DNA in the between, and then you can essentially have a pooled library, and then these libraries can be exposed to different conditions, and you PCR the barcode again. So we have used this DUP-seq library or overexpression library for diverse conditions. I'm just showing a snapshot of the data here. This is the E. coli library expressed in E. coli, and that library is exposed to different conditions. So I'm showing you different conditions on the x-axis, which shows different antibiotics, metals, so there are some other salts and some crazy stress conditions. And what you're looking at is the heat map, and particularly focusing on the darker or a purple color, which gives you survivor fitness. So if you have a deletion of a particular gene, the bacteria survives. And then you ask the question, why does it survive? Then you go and do the following experiments to validate. I'm showing you a snapshot of the genome browser there, where x-axis is a genomic locus, and then y-axis is a score. The red are the fragments which survived nickel stress. And then we can quickly go and find out which gene is this. And it encodes a particular gene called RCNA, which is involved in nickel efflux pump. And you can see that when I express this pump, it gives a resistance to nickel stress. So these kind of data sets quickly starts giving, associate the genes to a function. So how do we use these technologies for phages and understanding how the phage interactions mechanisms work and what are the rules of infectivity pathways? So we used to have a proof of concept. This is a couple of years, 2 years back now, published in PLOS Bio. Essentially, we took 3D technologies, and then we thought, okay, then let's take 14 different phages in E. coli. These are well-studied phages. In fact, each of these phages have a chapter in a phage bacteriophage book. There's a lot of information available. So we thought, okay, let's use these because there's so much is known. In fact, everything is known. So can we find them any new things, or at least recapitulate what is known? So experiment is simple. We take these pool libraries, and then you add phage, different multiplicity of infection, and then you collect the survivors, and then you check the barcodes who survived this condition, and then start understanding the receptors if it is possible. So this is a snapshot of data, so don't get boggled down by the information here, but I'm just x-axis is different phages at different concentration, y-axis is different genes. What we are looking at is a purple bar or a purple dark color, which gives a highest score. The yellow squares are the known data, so we validate what is known. So just to take up an example, for example, phage T4 here, it's here. The receptor phage T4 uses is OMC. So if I delete OMC, is a porin, the phage cannot get in because phage needs OMC because phage releases the DNA through OMC. So if you delete a receptor, the bacteria survives, and you can quickly use this kind of screens to identify not only receptors, but also enzymes, which are important for other functions for a phage replication and so on. So this kind of a data gives quickly a fitness phenotype for a particular phage. So it also gives a resolution at a LPS. LPS sugars are important, phages bind to LPS, and then it's always difficult to understand which gene is essential, which sugar moiety is important for phage infection. These technologies can quickly give you that resolution of possibility. So this is a gain of function technology where I'm expressing E. coli in E. coli fragments in E. coli and then exposing that to phage. And here you can see that one of the genes particularly giving resistance to all the phages. And then, there are some other dark color. There's a lot of data possible, but this particular gene is involved in a production of a cholanic acid. Essentially, when you overexpress this gene, E. coli forms nice gooey structure around it. And so it interferes with the phage infection. And then the second point here is these kind of new genes. So gene phage N4 is like a 50-year-old. People have studied over decades now. In fact, we thought there is completely everything is known about this phage. Recently, it's realized that this is not unknown. More studies are coming out. So in fact, these genes are involved in degradation of cyclic di-GMP, which is involved in virulence, pathogenicity, you name it. This is a second signaling mechanism. And now it is known that, in fact, this phage binds to a different new LPS, EPS, exopolysaccharide, and the cyclic di-GMP is involved in the regulation of the phage receptor. So these kind of studies definitely give more information on that. So we followed up these studies with some of the validations to do whether overexpression of one particular gene, which is unknown function, or overexpression of some of these degradation enzymes which are involved in phage receptors. So these results definitely, in fact, it gives us a more economical way of quickly doing the screenings and understanding the mechanisms of infection. So what next, right? This is the E. coli study. Can we do it for non-model organisms? And that was the main focus of these technologies. So since then, we have extended to Salmonella strains. Obviously, these are all model strains still. We have published Salmonella strain. Rest of these are all either soil microbes, ocean, and also human gut microbiome and strain from human gut microbiome. And one of the things that we had to struggle with isolation of phages. Of course, you can isolate phages from wastewater, but if you're isolating from a groundwater, the challenges are totally different. So we had to develop some different ways of isolating different phages for these kind of microbes. We have also gone one level up. Can we really expedite more than this? So we have, obviously, as any lab would do, we extended to droplet technologies. Now each droplet has a barcoded bacterial strain, and each droplet will also have a phage. So you have a very one-on-one kind of an interaction within the droplet. After incubation, you break the droplet, you collect the survivors, and do the bar-seq. And these kind of experiments are so rapid that we need to imagine what can we do with these kind of technologies next. We have also extended to something which we had never thought of doing it, is trying to understand how the phage infection is impacted by the metal conditions, abiotic condition, in this case, alkali metal conditions. And these ones, Hans Carlson has a scientist in Albino. He has carried out this crazy experiment where we have more than 80 different inorganic ions and how they impact phage infection. In fact, we see that in absence of phage, the IC50s are lower. That means phage gives protection to bacteria in extreme environmental conditions. We're trying to untangle that in a follow-up studies, but this gives the scale of things can be done. So where we are going with this. We have the phage resistance landscape or fitness landscape, we can do the similar things for different conditions. This I'm showing for different antibiotics, but you can do it for metals, preservatives, pesticides. And now once you have these kind of landscape, then you can think about putting that together. And then you can start one on the other, and then start trying to predict, can we see the trade-offs when the phage resistance happens? What are these other things give me sensitivity, or the other way around? When this is sensitive, can I have a more phage resistance, and can we use it for detection tools? So quickly cover the summary on this, that obviously we can do these all technologies, not in all the microbes. We need to have genetic tools for that bacteria. Then we can use that for making libraries and studying them. And then obviously gives the infection. Most important thing is this kind of data gives resolution for predictive model building, so that we can start building these phage-host interactions at a level which is not possible currently. The second quick snippet is on the phage functional roles. One of the things that you have heard this, the diversity of phages is so huge that we can't really comprehend how much information is there. So one of the example is a single-strand RNA and DNA phages. The number is, I'm completely lost the numbers now. I think more than 20,000 different single-strand RNA phages are assembled. We don't even know who they infect. We don't even know what do they do. But so mechanism of action is important, but also one of the key unknown in this case, the location of this lysis gene. These are 3 gene phages, very small, 3 to 5 kb gene. And the lysis gene is all over the place. So there is no real fixed location of this lysis gene. So there is a maturity protein, port protein, and the RNA-dependent RNA polymerase. So one of the key thing about this is lysis protein is super important, because imagine these 20,000 different phages infecting bacteria, and they must be targeting something which is different from DNA phages. DNA phages have more than 4 to 5 different proteins involved in lysis, whereas the single-strand RNA phages, one protein carries that function. So they are also called as protein antibiotics. The way they do this function is because of interfering with the peptidoglycan biosynthesis. I'm showing an example of this levivirus M here, which interferes with the lipid 2 flippase called merge. This, if you interfere this, the bacteria completely dies. And if by identifying these kind of targets, we can start thinking about protein antibiotics, which can be useful for the next generation. So this work is in collaboration with Adam and Ryan, who Ryan is a eminent scientist in this field, particularly single-strand RNA phages and lysis systems. And Ben Adelt is a grad student in UC Berkeley and is now postdoc in Doudna Lab. Here we took 6 different single-strand RNA phages. These are well-studied phages, but we still do not know what these lysis genes target to. We took these lysis genes and then we cloned them in a vector. And when we express this lysis gene, cell dies. In presence of DUP-seq library, which is an overexpression library, we can collect the survivors. So what happens here is you are inducing the lysis gene, and then you are delivering a suppressor possibility. And you can collect the survivors, and you can check who survived this particular lysis gene, and you can quickly map the resistance phenotype there. So this data gives one of the satisfactory thing is identifying, okay, this single gene protein M from a lysis M, it particularly gives suppressor of merging. And we found out a suite of genes which are involved in this kind of roles. Particularly is this PP7, which is a Pseudomonas aeruginosa phage. And this particular lysis gene also targets merge, which is interesting because these two PP7 and M are completely different sequence-wise. They do not match sequence-wise, but the target is similar. So this kind of a convergence of functional conversion of a particular target is interesting. And we might have to ask this question: if there are 20,000 different single-strand RNA phages or more, they must be targeting something which is on the peptidoglycan or outer membrane. So there must be a functional convergence in all of them. And this would be interesting study coming up next. And the final thing I wanted to touch upon is how do we map these functions of genes and what are the infection mechanisms are impossible, and asking this question, can we use them for applications? So these are the well-studied phages in E. coli, Lambda and T4. They have decades of work, hundreds of thousands of people worked on it, mapping each of them, at least some of them, what are the genes, functions of them, and still we do not have a complete picture of these. But these kind of approaches are awesome, but we cannot, if thinking about the number of phages we have discovering every day, can we really think about something more scalable? And why do we need these kind of approaches? I've just listed some of them. Conditional essentiality is pretty cool question to think about. If you're thinking of a genomic organization, why are things organized this way? Or can we have a minimal genome? Can we use it for applications? What are the host compensating genes? And how do we design these things better such that we can use it for diagnostics or human health or animal health, you name it, right? So one of the things that we started with was, obviously this field has gone crazy with number of CRISPR systems coming up. You have 9, 12, 13, I lost the number, but essentially 9 and 12 target DNA dependent, and whereas DNA targeting CRISPR systems, Cas13 is RNA one. Dinesh was a postdoc in the lab, and Madeline was a tech in the lab who developed this method where Cas12 in a plasmid, a dCas12, which is a catalytically inactive Cas12, with the guide RNA targeting different genes on a phage, can give a simple screen of, if the gene is essential, you will not get any plaque because the phage is going to, collapse. If the gene is not essential for phage infection, and if you target with CRISPR, you will see the plaque. So essentially, this gives a simple yes and no kind of a mapping. So we wanted to see whether this works well. So we chose 2 different phages, which we have enough data, which can validate things are working okay and nothing is crazy. So we chose Lambda and P1 phages. We have about 67 genes, and then P1 has about 117 genes encoding proteins. So here we wanted to confirm, okay, these things work fine. So we tried with a couple of major capsid. Protein is the essential gene for a phage. If you don't have a capsid, phage does not form. So Lambda has a E and no, these are essential genes for Lambda. So if you target them, phage cannot replicate well. Whereas P1 has got capsid protein packaging and then other proteins that are essential. If you target them, P1 cannot be successfully formed. Whereas these are the unessential, non-essential, like the proteins, which are called protein phosphatase and some unknown function genes. So we are satisfied with this. We wanted to scale it up. So, having a design well-known and having a Doudna lab next door helps much of many things. So for example, we knew the PAM sites for this. We designed guide RNA for each gene for each of these phages, cloned them in, and then transformed it to a bacterial strain. And then you expose it to the different concentration of phage, and then you collect the survivors or check the plaques and essentially calculate. So the following figure shows months of work mapping each gene, what is in triplicates, whether they are essential, non-essential. So I'm showing you just a summary data, which is red one is essential genes, whereas the green ones are non-essential genes, and some of them are not tested because they didn't have PAM site on them. So overall, out of 67, we have about 61 targeted. Our data matches more than 90% of them because we have the data which helps in validating some of them. And there are some ribosome profiling data shows that it has got another 55 genes, which we didn't target in this study. This is the P1 phage, which organization is totally different, as you can see. There are small patches of non-essentiality genes and some of the essential genes. And this kind of a data is super cool because you can start understanding which genes are important, but not super essential, not essential, and those kind of things. And also, this data is interesting. Out of 117, we see that about more than 95% of the data that we have with a single guide RNA, we will be able to map with the essentiality of this phage. So what do we do with this kind of mapping, right? The next thing is what is the use of these kind of mapping and how do we use it applications? So I'm showing 3 different snippets we have followed up, but I'm just going to touch upon one of them. Is we can use the non-essential parts for doing interesting things. You can put your interesting payload. We have used barcoded phages. We insert a barcode so that we can track them in different conditions. We can put different genes which can cause burden for a cell, or we can use it for more functional genomics approaches, complementation, and so on and so forth. So barcode phages, just touch upon how do we use it for barcoded phages. Imagine having a patient pool and we want to treat them with different phages. You can have a different barcoded phages. You can change the formulation without any issue because your downstream processing is going to be similar. You can barcode PCR, quantify which barcodes are performing well, which barcodes are not performing well. You can quickly start addressing quality control, whether it is following which phages to track, how the treatment efficiency is, what is the bioavailability of phages. You don't have to have design primers for every phage that you're thinking of using. So we use that for P1 phages and lambda phage. We have done some follow-up studies with pair of them. Seems reasonable. We're following in publication for this in a couple of months. Just to summarize this part, CRISPR-I definitely enables this kind of rapid, quick screen or assay identifying gene essentiality. We can, use this genome data for a lot of interesting studies, whether it is a fitness or a, functional analysis of this. One of the loopholes of CRISPR-I is the polar operon effects. Imagine the downstream gene is essential. So if you're shutting down the upstream gene, then the screen is not going to work. So having that kind of an, information at the prior would be helpful, but otherwise you should always critically check what data is there. Obviously, we have not screened all the design parameters. There's so much to be done in terms of design space there. But we are exploring a couple of these options. Barcoding definitely helps in standardizing the workflow, or also quality controlling your phage stocks in the lab or in the hospital and so forth. But the real star are these guys. These guys are the real threat to the next generation. Some of these strains are really XDR. They cannot be treated with any antibiotic out there. Whether you look at the nature, whether it is in a Pakistan or for India or UK, some of these strains are extremely resistant to all the antibiotics out there. So single labs doing any of these phage therapy or any of these things is awesome initiative, but it's not going to scale. What we need is a really holistic treatment, entire community coming together. So we put together this white paper in iScience in this year about the Phage Foundry format. I think we need a more systematic approach. If you're thinking of this whole phage community is not some bubble, everything is being shared. We know that, we saw that today in the morning, that things are being shared. I mean, imagine these antibiotic resistance genes passing through all the things that we handle, or whether it is the hospitals or industrial meat production or food production. So the things are going to be pretty nasty if we're thinking of bioterrorism agents and so on and so forth. So we need to be ready for, it's not like a sales pitch or anything. It's a reality now. So we put together this systematic workflow, and it's a huge goal. If we have a systematic surveillance of the environments, and then we can have the more systematic phage and host characterization facility. Not all the labs will have the technology or the money or the power or facility to carry out these experiments. So you need a more foundational facility where these can be done, whether it is characterization, engineering, and so forth. You need more knowledge-based sharing. Right now, there is literally less, other than sequence data, literally nothing is being shared between phage researchers, for example. So we need this Phage Foundry knowledge base where things are stored or at least shareable so that people can learn and understand from each other, so that we can really start making rational cocktails, engineered phages for a variety of applications, and so on and so forth. So take a look at this. This is not a finished product, it's evolving. And obviously this is just a white paper for, stimulate the field. So we call upon, really, we need more systematic surveillance. We need sampling isolation methods. We need more advanced characterization. We need long-term investments. It can't be less one patient or one facility or one application. We need much more stronger, full, look at the whole road. Anyway, so many people to thank. I just want to blue names in blue letters are the people who are directly involved in most of the data that's generated by them. And then I'm thankful to all the collaborators, and thanks for your time.
Yeah, this is actually a small question about the droplet experiments.
Yeah.
How are you getting single phage particles into droplets?
Oh, yeah, no, single phages are not going to be controllable, but bacterial strains are. We have a single barcoded, single or more than 1 or 0 or 4 bacterial strains. And phages are going to be at different dilutions. Obviously, we can't control the number of phages per droplet.
So you're using like a probability distribution.
Yeah, exactly.
Got it.
So we think the range is more than 100 MOI.
Okay.
Like one bacteria versus hundreds of phages.
Got it.
Obviously, we tried different dilutions, but that much more, 100 is more than better.
Okay, thanks.